All right, so we're going to start off, we're going to start calling DNA and all its associated proteins chromatin. So we have our DNA double helix, but that doesn't exist in isolation in the cell. It's generally associated with lots of other proteins. I'm going to look at a few in the next few slides. But all of that together is called chromatin. So DNA and all the proteins it's bound to. We had a few clicker questions on Friday to start getting you thinking about how DNA is packaged. It's packaged, if you look at this micrograph, so it looks like beads on a string. That's DNA wrapped around this core of 8 proteins called histones. Each bead is called a nucleosome, and it consists of a structure of that DNA wrapping around a set of 8 histones. There's an additional histone called a linker histone, H1, which clamps down the DNA at the base of that nucleosome structure and keeps it stable. That set of 8 histones consists of 2 of each of 4 different types of histone proteins. So 2H2A, 2H2B, 2H3, 2H4. So 8 proteins, 4 sets of 2 go into that octamer. Just to give you a little bit better idea of what those proteins actually look like, H3 and H4 form a dimer, so 2 proteins interacting. And then they interact with another dimer to form a tetramer. The same thing for H2A and H2B, right? So they interact in very specific ways, and then they all come together to form that octamer. We also had a clicker question on Friday about how much DNA it takes to wrap around that core histone, set of histones. How many?
Approximately 150.
Approximately 150 base pairs to wrap around that core twice. The structured DNA between the nucleosomes, the linker DNA. You want to guess how many base pairs there is in that linker stretch? Wild guess. Good guess. It's almost like you knew the answer. So it's 50 base pairs in that linker stretch of DNA. All right, so think about how many nucleosomes there are. Think about the thousands and thousands of base pairs there are in a chromosome. You've got nucleosomes essentially every 200 base pairs. 150 wrapped around the histones, 50 in between. Right? That's a lot of structure. Also notice our nucleosomes, they're about 10 nanometers across. All right, so we're going to start paying attention to the size of these things to figure out how we get them packed into the nucleus. So we started off with our DNA double helix. We should all be super familiar now with the diameter of that, 2 nanometers. All right, so if we just have our double helix stretched out, we know that it doesn't exist that way in the cell, right? It's wrapped around these sets of histones for stability and packing, with those beads, those nucleosomes being about 10 nanometers in diameter. That structure, that bead on a string structure, then gets coiled up into a strand of rope-like DNA. So a chromatin fiber. We're now at about 30 nanometers of diameter. Then we can take that chromatin fiber and begin to pack that together. We can pack it together loosely as these looped domains. So just loop it back and forth. Or we can coil it up even tighter. So the degree to which that chromatin fiber is packed together. We have two different terms to describe that. Heterochromatin, tightly packed chromatin. Euchromatin, loosely packed chromatin. Why do you think it matters how tightly packed the chromatin is? For mitosis. How so?
Chromatin gets packed together in chromosomes when undergoing interphase.
It gets packed together into chromosomes. So why would you want it really tightly packed in mitosis?
Because that's when you're separated.
Because you don't want to lose any of your DNA. You can imagine it's much easier to move around a tightly packed ball of stuff than if it's unwound in a string, trying to pull that apart. So there are certainly times that we want really tightly packed DNA. When might we want loosely packed DNA? In the gap phase. What's happening in the gap phase? DNA replication. Certainly easier to replicate DNA. It's not all bound up and wound around itself. What else? Normal cell function. What do cells do? I heard it. What was that? Reading the DNA. It's expressing genes. Proteins have to be able to get to that. Machinery has to be able to get to those genes to express a particular gene product. Right? So access to that DNA is important when you're trying to express things. So you want at least pieces of that DNA to be loosely wound so that the machinery can get access to it for DNA synthesis, for gene expression, anything you want to do with that information held in the DNA. All right, so we have our heterochromatin. All right, so we've squashed our DNA. We've wound it our nucleosomes. We bound that into a coil. Then we're coiling up that coil, packing it even tighter. So we get to that highly condensed, duplicated chromosome in a dividing cell. Right, as we just discussed, much easier to move around a tightly packaged thing as a unit. So we're going through any sort of cell division, we're trying to divide material up. We want it very tightly packed. So if we look at all of this put together, how we pack that together. So during cell division, the packing ratio for chromosomes is 15 to 20,000 times more condensed than a loose strand of DNA. That's really tightly packed. Why do we need it so tightly packed? Where does this have to fit inside? Nucleus. How big are nuclei generally? Anybody have any ideas? How many? 1,500 nanometers, good guess. A little bit on the large side. How many? 0.5. 25. Which unit?
Nanometers, sorry.
Nanometers? Think we're getting closer? Any other guesses? So nucleus on average is about 6 microns in diameter. All right, so we've been looking at all of our DNA packing in nanometers, so notice we switched units. 6 microns for an animal nucleus. It's much smaller than 2 inches of DNA. All right, so the nucleus takes up approximately 10% of the cell volume. So it's a big chunk of the real estate inside of a cell. But we still have to pack our DNA really tightly to get it in there. All right, so that leads us to thinking a little bit about cell organelles. So how well do you know your cell organelles? Show of hands, who's got the organelles down pat? I see sort of a halfway up there. Yeah? You at least know sort of what the cell organelles are, right?
Yeah.
Give me a list. What are some cell organelles?
Mitochondria.
Mitochondria, ribosome,
Nucleus.
ER, vacuole, Golgi,
Endoplasmic
lysosome, peroxisome. That's a good one. Chloroplast. What are chloroplasts only in? Plants. Endoplasmic reticulum.
The smooth end.
Smooth and rough. Have we got them all? Nucleus, we just covered. Nucleolus, very good. Peroxisome.
Cytoplasm.
Cytoplasm. Cell membrane. Anything else we need to add to our list?
I don't know.
We did get mitochondria. All right, so we know the list of organelles. How comfortable do you feel with the function of each? Not so much. All right, so we need to know all the functions, not only what they are, but what the functions of these organelles are. All right, so there's just a list. We actually came up with a more comprehensive list than this. All right, so I'm going to put a table up on Malima later today that has a list of organelles, and for you to fill in for each column, whether it's found in plants and animals, what its function is, and what its size is. All right, we're interested in relative sizes of things. All right, so let's see where you're at right now. A few quicker questions. All right, which cell organelles have their own DNA? All right, so this is just knowing something about the organelles.
Okay. Wait. Well, I mean, if we know. I go with you guys.
All right, everyone get their answers in?
Yeah, that's fine. Oh,
Take a look. Everyone get their answers in? All right. Excellent. Mitochondria and chloroplasts have their own DNA.
Oops. Let's see here.
All right, so what does that mean about their evolutionary history? Endosymbiosis. These organelles probably came from some long past symbiotic relationship with a prokaryotic cell. All right? So not only do you have to know the cell organelles, you have to know their function. So which cell organelle processes and packages proteins? Of that list, all right? Everyone get their answers in? Let's take a look.
Oh, God.
All right, excellent. Think you guys have organelles figured out for the most part? All right, so it is the Golgi. All All right, so here's the extra added layer for this class. Know your cell organelles. I think you guys are pretty good on those. You know their functions. Probably have most of those figured out. You know their size. How big are they relative to each other? All right, so on your own, for the first minute, I've given you a list of things in the cell. Rank them, order by size, smallest to largest. Think about all the images of cells and organelles you've seen. Looking at sizes of things in cells.
Okay. It like does a lot of stuff. Besides the oh, okay.
Everyone gets that list ranked. Best guess?
Oh, really? What is that? It's bigger than
All right, once you get your list put together, compare it to your neighbor, or a neighbor that you haven't already consulted on your list. See how close your two lists match.
Oh, and you know the ribosomes are bigger than DNA. Oh, yeah.
Well, no, I never put this in. I was just like writing the size. Yeah.
Is the central the thing? The thing that what's that second biggest?
I was going to think about that.
I feel like it's my breakfast is that's why I'm tired. Well, I think it's bigger than me, right?
I was just doing this.
I did too. I forgot.
In fact, I just read it.
All right. Let's see what you guys came up with. So let's start with the easy one. What's the smallest of that list? What's the largest of that list? All right. That's 2. 2 out of 6. What comes next? I've heard 2 answers, cell membrane or ribosome. What's the next largest set after that? Centriole. Which leaves? Mitochondria. All right, pretty good. All right, so there's the list with the average sizes. So toss up between cell membrane and ribosomes for some of you. Ribosomes a bit bigger than the thickness of the cell membrane anyway. All right, so I want you to have an idea of the relative sizes of things. So there are some things I expect you to know the actual size of, like the diameter of the double helix of DNA, the size of a nucleosome. All right, but I want you to have an idea of the relative sizes of the things in the cell. Right? It's important to understanding how things work together. Right? How thick is that cell membrane that keeps everything in relative to everything that's inside of it? We're going to move into mitosis next. And centriole will play a pretty critical role in that. How big is that relative to everything else that's going on? We'll get to the mitochondrion later in the semester. We talked about the nucleus, that 6 micron in diameter nucleus relative to the packing of the DNA. We're going to talk about microtubules in the next, probably on Wednesday, right? That was part of your homework. Understanding these structures that are pulling and pushing things around the cell. Okay, so have an idea of at least the relative sizes of the organelles, with a few specific highlights where, particularly in the homework, I've asked you to learn the size of that particular structure. All right, so I will put up a table on La Lima to help you organize your thoughts. I think hopefully most of you from previous courses, at some point you've had most of the organelles, whether they're in plants and animals and what their function is. Then you can also fill that in as far as their size, so you have an idea relatively what's going on there. All right, so I'll put the table up for you to fill in, and at the end of the week I'll post a filled-in table for you to compare to. Okay? Any questions about cell organelles? All right. So let's go back to that nucleus where we started. I'm thinking about DNA and its packing, heterochromatin, when it gets really tightly bound up for cell division. Let's start thinking about chromosomes and how we divide cells and divide that genetic material. So how many chromosomes are there in a diploid human cell? Which means you need to know what diploid is, right? You have to know something about human genetics as well. Okay, everyone get your answer in? Let's take a look. All right, so majority of you picked D. 46 chromosomes. That is correct. What does diploid mean? Two. There's 2 sets of the genetic material. All right? 46 means we have 23 chromosomes. In a diploid cell, we have 46, 2 copies of each. All right, what about in a diploid mouse cell? Take your best guess. Everyone get their answers in? Let's take a look. All right, so the majority went with 40, correct? All right, chromosomes in different species. Right, so if the mouse diploid chromosome number is 40, what's the haploid chromosome number? 20. All right, as we move into mitosis, and later on into meiosis, keeping track of diploid and haploid becomes important. All right, and particularly pay attention in exam questions about whether I'm asking for diploid or haploid. That changes what you're going to be calculating, right? All right. So that takes us into mitosis. And the chromosome mechanics, right? As we go through mitosis, we're really concerned with state of the chromosome, how many chromosomes there are, what they're doing in a particular mitosis phase. What happens to the chromosomes? And how do you describe the chromosomes in each phase of mitosis? Is really what you had to do for your homework, right? So there is a picture of a cell undergoing division. Yeah, whoa, right? You've got a bunch of green spaghetti stuff attached to a bunch of blue blobs. The green spaghetti stuff is our mitotic spindle. It's the microtubules that are going to pull apart our copies of the chromosomes and push apart the cells. The blue blobs, those are our condensed chromosomes. All right, and so we're going to look at each phase of mitosis about what's happening, particularly to the mitotic spindle and to the chromosomes. There are bunches and bunches of movies online that you can watch to help you with mitosis. I'll post these on Facebook. If you have a particular favorite, they'll be in your slides on La Lima. If you have a particular favorite movie, feel free to post it as well. The more you watch it, the better. All right, so let's look at mitosis. It's the process of cell division. We're going from one cell to two. Oops. Count the chromosomes. Describe their structure. How many chromosomes are shown there? 4. So this is a notation you're going to see from now on in lecture. So we have a stick of DNA with a blob in the middle to represent our chromosomes. What's the structure of the chromosome? How would you describe it? So the single copier has been duplicated. All right, what step are we missing in this process? We're going from 1 to 2. What am I not showing? Not showing the whole process of mitosis, but relative to the DNA, I'm showing you the DNA replication. All right, we're going from 1 copy of each to 2 copies. Somewhere in there they have to be duplicated. All right, so we have to, at some point in the cell cycle, duplicate our DNA. And cells that are dividing are continually in this cell cycle. Right? They have to not only divide, there's a period where they grow, gain resources, duplicate the proper things in the cell, grow some more, then they can divide. Then they start the whole process over again. So we have the G1 phase. So the cell cycle consists of all these 4 phases, which you should be familiar with. The G1 or GAP1 phase. So this is where the cell is growing, gaining resources. So you can imagine right after cell division, these cells are kind of small. They don't have a lot of resources. They put that all into the cell division process. All right, so we have the first gap phase, or G1. You get cell growth. Those cells get big enough. They enter the S phase, or DNA synthesis phase. So you're going to replicate your DNA at this point. You've got 2 copies of everything. You go to a second gap phase, or the G2 phase, where you do a little bit more growing and you make sure that you've duplicated everything properly. And then you can enter mitosis and do cell division. So we're going to talk a lot about that M phase of mitosis, where cell division actually happens. The remaining 3 phases, G1, S, and G2, are often referred to as interphase, so the phase between cell divisions, even though we've divided that up into 3 separate stages. So let's look at the cell cycle a little more closely relative to what's going on with the chromosome. So in that G1 phase, just after cell division, we've got a single copy of everything. Our chromosomes haven't been replicated. We have a single MTOC. What's MTOC stand for? Anyone know? Nope. So that stands for microtubule organizing center. So in that first image, the green spaghetti was coming out of a centralized region. All right, this is a structure, an organelle in the cell that organizes our microtubules for moving things around, particularly at cell divisions. All right, so in that G1 phase, chromosomes haven't been replicated. We have a single MTOC. We go into the S phase. What happens in the S phase? Synthesis. How would you describe the DNA afterwards? There's 2 of them. It's been replicated. It's been duplicated. What about the MTOC? What happens to it during the S phase? So think about in that first image with the green spaghetti. How many green spaghettis were there? So many. How many central bits was green spaghetti coming out of? 2. All right. So there have to be at least 2 centers organizing those microtubules. All right. So in that S phase, you don't only get DNA replication, you get replication of your MTOC. All right, so thinking about how we are referring to our DNA at different stages, particularly in these cell division processes, can get confusing. All right, we've got DNA, we've got chromatin, we've got chromosomes. So let's try to clarify it a little bit. So how many chromosomes do you see in this image? All right, we're after the S phase. How many chromosomes would you call that? All right, let's take a look. Ooh, even split. All right, so justify your answer. Someone who chose 4. Why did you choose 4? So the definition argument for 4, that one chromosome is made up of 2 chromatids. Anyone want to justify 8? Why did you choose 8? No one? There's 8 lines. That's a good reason, right? We're talking about duplicating chromosomes. There are 8 of them shown up there. So the answer should be 8. But here's the catch. All right? Chromosomes are separate entities. So after they've been duplicated, they're still attached by that centromere. That blob on the stick is the centromere of a chromosome. They're still attached, they're still one chromosome. Right? That's how things get a little bit tricky. So it's true that in each case, it's a single chromosome that's consisting of sister chromatids. So there are 2 copies of each chromosome, but they're still associated with each other. What's the diploid number? How many chromosomes are we dealing with unreplicated? So what's the diploid number? 4. Remember, there are 2 copies, right? Assume that we're talking about an animal, right? So if you look at these, so the proper one here is 4. Get rid of that. All right, so you see there's 2 forms of a chromosome that are long with the centromere in the middle. All right, so that's a pair. 2 that are short with the chromosome up top. That's a pair. So your diploid number here would be, haploid number would be 2. All right, so we're talking about cell division. We're not changing the number of chromosomes. Just checking in. We'll do that when we get to meiosis. All right, so you want to keep track of the state the DNA is in. Before it's replicated and after it's replicated, so that changes how you refer to it. All right, so every chromosome equals 1 continuous double-stranded DNA molecule. After they're duplicated, they're still attached at that centromere, so you still refer to them as 1 chromosome until they've been pulled apart in cell division. All right, so we look a little bit at the chromosomes. All right, so let's get into the actual phases of mitosis. And as you did for your homework, you have to know what state that DNA is in each phase. All right, phases are defined by what's going on with those chromosomes. The first thing, can you write down the phases of mitosis? All right, what are the phases? What's the order? Interphase. Prophase. Prometaphase.
Metaphase. Anaphase. Telophase.
And finally, cytokinesis. How do you remember the order? What? I haven't heard that one. What is it?
I pushed my Aunt Terry.
I pushed my Aunt Terry.
You see it from it. I pushed my Aunt.
Yep, that's a good one. I hadn't heard that one. Anyone else have a way to remember them?
IPP mat.
IPP mat. That also works. Get prometaphase in there. Any other good ways to remember the order? All right, so you have to know not only the order, you know what's coming next, right? Can you draw each phase like you did for your homework? Prophase, or metaphase. That's what we went over. All right, so you should be familiar with what happens in each of these phases, what they're defined by relative to the chromosome. So I think we'll stop a few minutes early today. We'll pick up on Wednesday, prophase, go over all the phases. We'll talk about microtubules and what the cellular machinery is that's moving these things around. All right, so think about that homework question. We're looking at microtubules and motors.